Very good morning to one and all. Today's topic for the lecture is preparation of gene library. Before understanding about the gene library, there are three words which we need to understand, that is genome, proteome and transcriptome. As we can see, each of the three words ends with the word ome, that comes from chromosome. So, genome is the combination of gene plus chromosome, that is the sum total of all gene present within the nucleus of a particular cell. Similarly, proteome means the sum total of protein content of a cell, and transcriptome means the transcriptional unit of a cell. Transcriptional unit means the all content of RNA including mRNA, tRNA and rRNA. After understanding about these three terms, we directly switch to gene library. What is a gene library? As the name suggests, gene and library. Library means the collection of something and gene library means the collection of genes. Gene library may be of two types, genome library and cDNA library. Genomic library comprises of the collection of Cdna, that is genomic DNA. The place or the content where you can see the sum total of a particular genome of a particular cell is referred to as genomic DNA library. How we can prepare the genomic DNA library? There are few steps which are need to be followed in the preparation of genomic DNA library. In order to prepare gene library, we have to first isolate the DNA content. So, isolation. After isolation, we have to load it into a particular type of a vector. After loading into a vector, we need to check out the cloning or the expression of the loaded DNA. For isolation, we need to rupture a particular cell type from which we are isolating the DNA. Isolation could be done with the help of either sonication. Sonication means the mechanical disrupting of the membrane of a cell either by mechanical means or with the help of a particular type of detergent. We have isolated a particular DNA content of the cell. After isolation, we need to cleave it into small fragments. This cleavage can be done with the help of restriction endonucleases. Now, what are restriction endonucleases? As the name suggests, restriction endonucleases. Endonucleases are those enzymes which cleave the DNA or RNA or any particular type of biomolecule within the particular site of the biomolecule. Restriction endonucleases are those endonucleases which cleave the DNA at a particular site, the site being called as recognition site or the restriction site. After cleaving the genomic DNA, we have isolated into smaller fragments. We straight away move on to the Agarose Gel Electrophoresis. Agarose Gel Electrophoresis means the separation of the isolated or the fragmented DNA according to their size. After following the Agarose Gel Electrophoresis, we load these into a particular type of vector. Now, what is a vector? Vector is a particular type of molecular vehicle in which we can load our foreign DNA in order to transfer it from one organism to the host organism. After loading into the vector, we transfer this molecule into the host. Now, how can we load it into a particular type of vector? We can transfer these restricted fragments of DNA into a vector molecule with the help of enzyme called ligase. What is a ligase? Ligase is the molecular glue. Molecular glue means the type of a enzyme which helps in the sticking of a particular type of biomolecule to another type of a biomolecule. Similarly, restriction endonucleases, since they are being cleaving at a particular site, they are commonly known as molecular scissors. After you load it into a host molecule, the type of a vector molecule having the foreign DNA starts recapitulating itself and we obtain the particular type of gene library. Gene library has its advantages as well as its disadvantages. Amongst the advantages, it includes for the sequencing of a genome, we cannot sequence any part of a genome until and unless we have the particular type of the gene library. This is about the gene library. Another type of the library is the cDNA library. What is a cDNA? cDNA means complementary DNA. Complementary DNA means complementary of what? Complementary of mRNA. Means in genomic library we were using the gDNA. Now in cDNA we are using the mRNA. How can this cDNA library could be prepared? For the preparation of cDNA, as we all know that cDNA is obtained from mRNA, we need to isolate a particular type of mRNA from a particular type of tissue or the cell. So first we isolate the mRNA. Then after isolation of mRNA, we need to convert it into a cDNA. And after converting mRNA into cDNA, we need to prepare the library of cDNA. Library of cDNA is just we isolate a particular mRNA, convert it into a cDNA and then load it into a vector for the replication of the mRNA. So mRNA being translated into cDNA. So how could we convert mRNA into cDNA? First, we all know that mRNA contains a poly A tail at the 3' end. Now we have to convert this mRNA into cDNA. We all know that DNA converted to RNA is referred to as transcription, and now we need to convert RNA into cDNA. So the process is called reverse transcription, and the enzyme used for conversion of RNA to cDNA is reverse transcriptase. So mRNA is having a poly A tail at the 3' end. So we can use a primer having poly T end at the 5' end. When this primer gets ligated to the 3' end, in the presence of reverse transcriptase enzyme, we have a one strand of mRNA and another strand of cDNA. Now we only need our cDNA copy. So what will we do? We will digest the mRNA strand with the help of RNase enzyme. RNase dissolves the RNA copy of the cDNA and RNA hybrid. After that we receive the single strand of cDNA. Now we need to convert this strand into double stranded cDNA. So we will use polymerase enzyme. We will use polymerase enzyme to convert this cDNA copy, single stranded copy, into double stranded. Polymerase enzyme will form a loop in the cDNA copy. Now we do not want any such type of loop, since it may hinder in the action of cDNA library preparation. Now what we will do? We will dissolve this loop with the help of enzyme S1 nuclease or any site specific nuclease. What nuclease enzyme will do? It will dissolve the loop and hence we will obtain the double stranded cDNA. This is about the preparation of cDNA. Now we want the cDNA library. What we will do? We will load this cDNA into a particular type of vector, and this vector being loaded into a particular type of host for the cloning purpose. One problem in the cDNA library preparation is this last situation is very rare, means we do not obtain the complete copy of the cDNA. Why is it so? It is so because the enzymes being used in cDNA library, that is reverse transcriptase enzyme, next RNase, and that is S1 nuclease. There may occur the problem in the specificity of these enzymes. Reverse transcriptase enzyme may sometimes fail to copy the complete mRNA into complete cDNA. If our reverse transcriptase is properly working, there may occur the problem in the RNAs. RNAs may fail to dissolve the complete strand of mRNA in the mRNA and cDNA hybrid. And there may also occur the problem in the specificity of S1 nuclease. It may fail to dissolve completely the loop of the mRNA so formed. So there are three problems which we can face in the preparation of cDNA library. Now we have known about the gene library and cDNA library, how we can screen out the gene library and cDNA library. So prepared. Screening means we have prepared the genomic library and we have prepared the cDNA library, but we do not know that actually our DNA has been transferred to gene library or to the cDNA library. We need to know that in which part of the cell or in which part the recombinant DNA has transferred itself. So this procedure of knowing about the transfer of a DNA, rDNA means recombinant DNA or chimera. In which part the recombinant DNA has transferred itself, we will study in the screening. Screening could be done in two ways, either sequence dependent or expression dependent. Sequence dependent means we need to know about the sequence for which we are being screening out the library. In this we mainly study about colony hybridization. And expression dependent means we need to know about the expression of the transferred rDNA into the cell. Expression dependent में we usually study, there are a number of type of screening. Since we have to study in the gist, so we will mainly focus on colony hybridization and the hybrid arrested translation, that is HART. First we will move up on the colony hybridization. Hybridization, we all know what is the hybridization, means to hybridize or to make a mixture of any two or more than two components. Colony hybridization mein what we do, we make a plate, petri plate having a particular type of the growth of microorganism. Then the first plate is known as master plate. From this master plate we produce the replicas of this master plate. The replicas means we will touch a velvet sponge over this, and then same velvet sponge from this master plate is being touched to the another petri plate. So it will have the same number of colonies at the same place the master plate is having. This will be known as master plate, and the plate so obtained from master plate will be known as replica. Replica or the replica plate. After replica plating, we transfer these colonies to nitrocellulose filter membrane. After transferring to nitrocellulose filter membrane, since we know the sequence of the transferred recombinant DNA, we prepare the artificially the probe complementary to the sequence of rDNA. The probe will hybridize with the complementary DNA, complementary sequence of the DNA. Then after probe hybridization, nitrocellulose membrane is baked at about 60 degree. After baking at 60 degree centigrade, since we are focusing only about the recombinant DNA, so main aim for the colony hybridization is to isolate the DNA and to degrade another biomolecules including RNA or proteins. So we will digest the proteins with the help of proteinase. After proteinase, this hybridized probe with the known sequence is being autoradiographed. After autoradiographing, we come to know about the hybridization of the probe with the particular type of DNA. This is all about colony hybridization. Next we move on to the hybrid arrested selection. Hybrid arrested selection. As the name suggests, hybrid arrested translation means we are arresting the translation of a particular type of hybrid. See, what we study in biology or in biotechnology, the meaning is hidden in their respected words. For example, hybrid arrested translation. As the name suggests, that the translation is being arrested with the help of hybrid. Hybrid here is the hybrid of mRNA and DNA. What we do, we isolate the particular type of mRNA from the particular type of from the particular type of cell. mRNA being isolated, it is divided into particular types of lots. This mRNA will be converted into complementary cDNA, and the screening of the cDNA which is being tested by us will be transferred to the mixture from the external source. This cDNA will hybridize only with its complementary mRNA. Then we attain the hybrid of mRNA and cDNA. After attaining the hybrid of mRNA and cDNA, we transfer this to electrophoresis. Electrophoresis में what we will obtain the particular type of band. But in those situations in which mRNA is hybridized with the cDNA, there occurs no expression of mRNA. क्यों? Because mRNA is also being binded to the particular type of cDNA. So we have arrested the translation with the help of hybrid of cDNA and mRNA. In cases in which bands are missing, we can identify the particular type of cDNA and RNA. This is all about the preparation of gene library, and in screening there are a number of screening techniques such as colony hybridization, PCR, FACS, HART, HRT. A number of screening techniques are available. Last but not the least, we will study about the difference between the gene library, genomic library and the cDNA library. Since genomic library is being prepared with the gDNA, it may consist of both the introns and the exons part. Introns and exons parts are the coding and non-coding regions respectively of the DNA. But since cDNA is being obtained from mRNA, we do not have any non-coding region in the cDNA. That's all.